Okay, hi there. This is going to be part one of several parts on a lecture on for distance learning on protein synthesis and some other topics involved. But so I'll jump right in. Just a little bit of background. A genome is a term that refers to one complete amount of an organism's DNA. Okay, so for example, in humans we have 46 chromosomes or pieces of DNA per cell, and all 46 of those chromosomes together would make up your entire genome. Okay, so every cell in your body that's a body cell or a somatic cell, which would include hair follicle cell, skin cell, lung cell, kidney cell, ovary cell, muscle cell, any one of those regular cells that you have actually has a complete set of your genome. We could clone you from just one cell. The only types of cells that do not have a full set of DNA in you would be sperm or egg. Okay, they have half of your DNA in it, and that's because they're going to combine with another sex cell to make a new human zygote, it's called. But most of your cells have your full DNA in it. Now, the Human Genome Project was completed in 2003. It took a handful of people and actually mapped, using these complex machines, it figured out every A, T, and C, and G in the right sequence, all the DNA in the right sequences for a handful of people. And since then, the technology's gotten better, and we can do it pretty quickly now. Down here, this is DNA, okay, and segments of DNA called genes code for proteins, okay, and they give us our traits. So just to remember here, DNA codes for only one type of molecule, proteins, and those proteins give us our traits: our hair color, our eye color, our gender, our ability to metabolize certain foods, the efficiency at which we break down certain toxins, all sorts of stuff. Okay, now here are the 46 chromosomes in one human cell. This diagram is called a karyotype. Okay, kary means nucleus. Remember, eukaryote, kary means nucleus. And here's 46 chromosomes, and this is not something the cell arranges like this. This is something a scientist has to arrange. Scientists actually painstakingly, they do these, thousands of these are done, per year, for various reasons when they have to kind of do genetic testing and stuff. But they're paired up because these are two homologous chromosomes. One of these came from the mom, and one of these came from the dad. Okay, one of these came from this individual's mom, and one came from the dad. All right, that's why they have similar bands on them, because homologous chromosome means chromosome that has genes for the same traits on them. They're not identical genetically. For example, this little band, this third little black band up, and this third little black band is a couple of different genes, or maybe more, who knows, but it may code for, let's just make something up, eye color, right? So maybe mom's genes for eye color over here gave this person the ability to have brown eyes, and this one is from dad, and it has the blue eye gene in it. So they aren't necessarily the same, but they have the same information on them. Okay, karyotypes arrange chromosomes from long to short, and then there's a special set of chromosomes at the end that a scientist put here called these sex chromosomes. Okay, in humans and many animals, if you have an X and a Y, you are a, guess, male. And if you have an X and an X, you are a female. Okay? Here's another one, but take a look at this one. This one is a human one. It has 46 chromosomes, or 23 pairs, but it has 2 X's and no Y's. Okay, so it is a female. Let me just show you another interesting one. Here's a karyotype that is very much the same in a bunch of other ways, but it has 2 X's and a Y. Yes, humans sometimes have this happen. It's called Klinefelter syndrome, and they actually have both X's and a Y in every cell, and it has certain symptoms associated with it, but I'm just showing you some different examples here. Okay, now, I want to start with a scenario as we move into protein synthesis. And if I go back here, we're going to be talking about this topic, the intricate details of how a gene makes a protein. Okay, and that's called protein synthesis. But before we do, I just want to make an analogy, and I'm going to refer back to this analogy as we continue. So, one, let's pretend the school's power, let's pretend we're back at school, right? You're in your classroom. The school's power goes out, all right? And the reason I wrote this is just so that you can't use the phone, for example, and you can't use internet, okay? But your teacher has to get an important message during class to the principal's office. Okay, how do you get the message down there? I mean, you don't have to be a genius to figure this out. You send a student. The teacher sends a student with the paper, with the message on paper. Okay, why? Well, the teacher's quote unquote stuck in the classroom. Legally, the teacher can't just leave the classroom with the kids in there. And the information has to go from that classroom to somewhere else. So you have to send a messenger. All right, now I'm going to refer to this. I'm going to refer to this analogy as we go on because this is very much like one part of protein synthesis. All right, let's recall real quick. A gene is a segment of DNA that codes for a protein. The conventional thought is there's like 23,000 genes. I've read more recently that there's a different number more, but we used to say there's 23,000 genes per human genome. Okay, they code for proteins. Proteins have jobs like breaking down your food and building and repairing cells and all sorts of stuff. And their jobs are determined by their shape, just like a tool's job is determined by its shape, right? You wouldn't use a hammer to try to screw in a nail. Proteins are made of amino acids, and that's going to be important too. Remember, the building blocks of proteins are amino acids. How are they made? That is the process of protein synthesis. Synthesis is a fancy word to put together. So how are proteins put together? In 2 steps. First step is called transcription. Second step is called translation. Protein synthesis is really synonymous with gene expression. If you express a gene, you're actually making the product that gene codes for. Okay, it's very much like the DNA is like the blueprint and the protein is like the house, okay? Or the DNA is like the recipe in a recipe book, and the protein is like a meal, the thing that it has directions to make. Okay, now, what is RNA? RNA is very similar to DNA. It is a nucleic acid, just like DNA. RNA stands for ribonucleic acid because ribose is the sugar in the nitrogenous bases of RNA. Deoxyribose, which has 1 less oxygen, is the sugar in DNA. That actually makes RNA a little bit less stable, a little bit more likely to mutate, which is probably why DNA evolved as our hereditary material, because it's more stable. Okay, so the other thing you'll notice, this is DNA, and you should all recognize this by now, with the C's and G's and A's and T's in it, right? Look at RNA. It looks like it's missing half of it because it's single-stranded. It's half of the spiral staircase. And this is 2 halves of the spiral staircase. So we say there's 2 strands in every DNA molecule. Well, there's 1 strand in every RNA molecule. The other thing you might notice here is that we have the letters C, G, A, and T in DNA. Cytosine, guanine, adenine, and thymine. Look at what letters we have over here. RNA bases include cytosine, guanine, adenine, and not thymine, but uracil. It's slightly different. Over here on thymine, there's a little CH3 bunch of atoms called a methyl group, and that's missing on the uracil. Okay, so there is no thymine. If you see a U, you're talking about RNA. If you see a T in the sequence, you're talking about DNA. All right, so we're going to stop there. That's kind of a little background, and I will upload part 2, or I'll make part 2 and then upload part 2, which will describe transcription and probably get a little bit into translation. Okay.